Have you ever played the game Twister? You know, the one where you fold your body into weird shapes depending on the spin of a spinner? Well, if you have, then you know that there are some positions much more comfortable than others. And in our bodies, proteins have the exact same feeling when they are being folded. This is the process that we're going to talk about today. Proteins are long chains of amino acids responsible for almost all major biological function within the human body. Proteins are formed through DNA transcription and translation. Here, a gene coding for a specific protein is copied into messenger RNA, which enters a ribosome. There, every group of 3 nitrogen bases known as codons are matched with a corresponding transfer RNA molecule and its amino acid. Each time the next tRNA finds its matching codon, the amino acid sequence grows, until finally a stop codon is reached, telling the ribosome that the chain has been fully assembled, thus completing the protein's primary structure. From here, special properties within the amino acids make the molecule play its own game of Twister. However, unlike in the party game, protein folding is not done at random. This was discovered by Cyrus Leventhal, who proved that even under extremely strict parameters, a protein could take over 1 billion years to fold properly if done so at random. Therefore, there must be some hidden force making the same protein fold the same way every single time. In fact, recent discoveries shed light on this issue, proposing a 2 or 3-step method for protein folding. First, the linear primary structure of the protein engages in intramolecular hydrogen bonding, creating a secondary structure. This happens either vertically, creating right-handed spirals known as alpha helices, or horizontally, creating pleated sheets known as beta sheets. Next, hydrophobic amino acids bury themselves deep within the protein in order to eliminate any contact with water. This is known as the hydrophobic effect and creates the tertiary structure of the protein. In most cases, the protein is finally functional and can act on its own. However, in some instances, multiple polypeptide chains must be combined together in order to form a functional protein. Once the protein has finished its folding through either of these levels, the protein has entered the native state, and it's finally able to perform all of the biological functions it was designed to do. However, if something goes wrong in this folding process, such as a mutation in the transcribed gene or a deviation from the native state, misfolding can occur. And this has been connected with diseases such as sickle cell anemia, cystic fibrosis, and has even been associated with over half of all human cancers. Thus, an understanding of how misfolding occurs can help the scientific community develop cures for these diseases and help make our future healthier than ever.